So one important bit what we can learn is how actually our organs develop, and more specifically, not just to observe how the organs develop, what we know by now, but to really understand what are the underlying molecular mechanisms. So which factors are actually involved in the whole process. The guiding principle, or the underlying basically logic of this, can be sort of summarized, or like a famous physicist, the Nobel Prize winner Richard Feynman actually nicely summarized it when he said that what I cannot create, I do not understand. That means if we really truly want to understand how a process works, in this case organ development, we should be also able to recreate it in vitro. So we recreate it in the dish in our labs. And that gives us a huge advantage if we can recreate that process, because then we can also analyze it in more detail using methods that we cannot use, for example, on embryos or on patients, etc. That essentially means that we try to recreate human development or human organ development in a dish, but also disease state in a dish. And that also means then we can then go on to develop new therapies that are much more targeted for a particular organ, in our case, for example, the heart, and that we also understand much better what, for example, the side effects can be, and also to mitigate for these side effects. So that, I think, would have a huge impact on our knowledge, and also how we do medicine in the future.